Thank you, Ian, for that kind introduction, and thank you for attending this afternoon session on a Friday, which is always what I call the graveyard shift. My challenge this afternoon is not just about talking to you about feeding the next generation, but also addressing and pitching this at a level so that my 10-year-old daughter, who's seated out there, and Professor Ian McKinnon, who's seated out here, is able to grasp and take something home from the messages that I have embellished in this here.
Easier for her.
Overall, this is the structure that I plan to maintain throughout this afternoon, really providing a brief introduction, talking about some of the global challenges that are associated with population dynamics, stress tolerance and how it fits into the entire scheme of water use management, talking about extremophiles and where I use a native Australian resurrection plant as a model system to develop strategies on how we could improve stress tolerance. Another passion of mine is about bridging this innovation chasm and why a place like QUT is such a fertile environment to help deliver such outcomes in helping to bridge, take R&D to the marketplace or to the people who actually need it. And to give you an example of a project that I'm currently involved in leading, and this is around helping to address the demand and the need for plant-based proteins in Asia, particularly the Indian subcontinent. I could not resist but show you this particular slide, which is an article that appeared in 1860 in the Natal Mercury, which is the main newspaper in a province called KwaZulu-Natal in South Africa, where my forefathers were brought across as indentured laborers to work on the sugarcane plantations. And the title of the article was "The Coolies Are Here." And the coolies is one of the most derogatory terms that you could use to refer to a person of Indian origin. That was the welcome that set the stage for a huge challenge that my forefathers had experienced, which is really what has made me so passionate about this field. And all that I'm trying to instill in you is that, as one of my favorite writers, Malcolm Gladwell, who writes for the New York Times, says, if you experience a challenge in your life, sometimes it puts this fire in your belly and it keeps you charged. Hence, being at work at 5 every day, as long as I can remember, and my wife, who's seated out there, would be testimony to that, is not because I have to be there, it's because I want to be there and I want to make a difference. And this slide here captures South Africa going from the cane fields, at least my forefathers, to freedom. Now, I returned from an agricultural innovation conference just a couple of weeks ago. Seated at this innovation conference, there were industry players, there was farmers represented, scientists, etc., trying to grapple and deal with the challenges that are actually facing mankind in terms of how do we sustain the future populations, particularly with the growing demand and growing populations, etc. So I wrote down all the terms that came to mind and phrases and concepts, and I gave it to 2 of my budding students, Paraj Kabashi and Grace Tan, and they put it into a word cloud, randomized it, and this is what came out. So it's no secret that population growth, land degradation, food security, drought, the use of a variety of technologies, climate change are some of the key drivers that are actually going to affect and are currently affecting our ability to be able to deliver the challenge to be able to feed the future generations, if not just the current. Now, I know amongst you out there, including in the political echelons of many countries, they are the non-believers. Is there climate change? Is it occurring, or is it just something that has been politicized or created such that people are actually making this into a very political concept? Now, we know that there's a lot of data, scientific data, and scientists who have actually shown that there's a dramatic change in the temperature of the earth, there's a dramatic change in rainfall patterns. You just got to look at the weather patterns that we're experiencing in Brisbane for you to know that. I was going to demonstrate this from some personal examples that I've had. Around 8 years ago, I climbed Mount Kilimanjaro. This is what Mount Kilimanjaro looked like as we were summiting. And this is what it looked like right at the top with this massive ice glacier. It's predicted that with the global climate changes that are occurring, particularly rises in temperature, by 2020, Mount Kilimanjaro will be bald. Now, that is a reality that we are currently experiencing. 3 years ago, and I've got two of my team members in the audience here, Brett Williams, my research associate and wife, accompanied me, and 7 other mates from across the world, and we spent 12 days, trekking in the Himalayas in a region called the Annapurna region. Annapurna in Sanskrit means crops in abundance. And it was fascinating to see that as we trekked through the farmlands, through the range in Nepal, the Himalaya range, there was a lot of subsistence farmers for hundreds of years had been dependent on agricultural activity. And you could see the strain because of all the changes that are occurring climatically in them being unable to kind of get the harvest that they were expecting. And you could clearly see that this is, in reality, they are going to be changing their patterns and even their diets in order to be able to accommodate the ability to be able to sustain themselves and survive. I was in the southern part of India, beginning of this year, but beginning of last year as well. This slide was taken when a colleague of mine, who is the head of the Department of Rice at Tamil Nadu Agricultural University, where we walked in the rice paddy field in January 2013, when the monsoon rains that were predicted and expected to arrive did not arrive. And this is what the paddy field looked like. And I took this photograph with my iPhone. What you do see here is the fact that there is no way, if the rainfalls didn't arrive, that this crop was going to result in a harvest. And that is what had happened. The dams were dry. So, in essence, what we are seeing is that there are tangible effects of climate change that's affecting livelihoods across the world. So this is not an enigma, but a reality. Now, the other challenge that we have is this massive dynamics in terms of population growth. And this is pressing and placing a huge pressure on the natural resources that's found. Now, if you just take a look at from 1960 through to 2000, the population increased from 3 billion to 6 billion, and in order to be able to sustain this, food production had to be doubled. If you take a look at the predicted growth of the population to 2050, in order to be able to sustain this 9 billion population, we need to increase food production by around 70%. Is this really going to be sustainable? We need to really be smarter about how we deal with agriculture, and I hope today to provide you with insights on how we not just look at food security, but look at food and nutritional security and how we could position ourselves to help deal with this. Clearly, one of the major limiting factors that's affecting productivity of crops is the abiotic stress, and I'll tell you more about this in a short while. This data comes from the United States Department of Agriculture. The reason I present it is because I think it symbolizes what's happening across the globe. We often have the misperception around the factors that actually affect crop losses. But taking a look at this pie chart here, you'll notice that close to 70% of agricultural productivity is actually dependent on water-related issues. Either there's not enough water or there's too much water in the case of flooding, causing anoxic stress and resulting in severe retardation of crop production. Now, if I haven't driven the message home already, the amount of available fresh water to us on this planet is actually quite minuscule. If you take a look at this, it's only 2.5% of all the water that's available on Earth that's actually fresh water. And of this here, 70% is actually locked in the Antarctic ice caps. And whatever is left and available to us, we are essentially utilizing a significant percentage of this in agricultural activity. And with the increasing demands that we, because of our behavior and patterns and how our lifestyles, because we start to put pressure on this access to this water, it's going to have a significant effect on our productivity in terms of crops. We need to look at better management practices. We need to look at strategies to improve and enhance resilience in crops in order to be able to ensure that we can better utilize this very limited and invaluable resource. So when I talk about abiotic stress, what I'm really referring to is a range of stresses. Drought is a significant component, but there's also salinity, nutrient stress, temperature stress associated with low as well as high temperature, and UV light. Plants are immotile or immobile or sessile. Unlike humans, if it's too warm outside, we move on to a sheltered area. Plants are unfortunately sedentary. They are exposed to all these conditions, and they got to enhance in some way or the other the ability to be able to tolerate it, failing which you will find that they will completely be annihilated, and I will talk more about it in a short while. Now, the way plants respond to the stress, or stresses in general, is a very complex process, and in no way am I going to boulderize it and simplify it and give you the misperception that it's just a very simple way of actually putting in a certain gene and expecting to create a stress-tolerant plant. From this, you'll notice that the stress needs to be perceived, and once it's perceived, there's a range of compounds, whether it's a plant hormone like abscisic acid or a range of transcription factors, ultimately resulting in changes in gene expression that results in tolerance. So if you could just think of this as being an orchestra of instruments, and all the genes being the instruments. You cannot play all these instruments all the time because it consumes so much of energy that the plant will not be able to sustain itself. So the conductor, which is really these transcription factors, will be able to say, for this particular melody, we need this type of tune, these instruments to be playing. For that type of melody, we'll need another bunch of instruments. And in doing so, we would be able to help enhance stress tolerance if we can get a better understanding of exactly who these conductors are and what exactly they are doing. I want to make a strong distinction between drought as well as desiccation, because this will then be a segue for me to introduce you to the resurrection plant that I work on. Most of our crops, in fact the majority of them, are drought sensitive, which means that if they lose 20% of their relative water content, they are already in panic mode. And they will have to make a conscious decision, do we hang in there or do we actually give up? And in most instances, if the relative water content drops down to about 70 or 60%, they are more than likely to say, well, time to give up. And they initiate what is called programmed cell death, where they consciously self-destruct. I'm going to introduce you to a group of plants that we call resurrection plants, and we study Tripogon loliformis, which is a native Australian resurrection grass, which can be dehydrated down to less than 5% relative water content. So rather than it sending out a message that it's too hard and too difficult to hang in there, it says hang in there. Water will arrive, and when conditions improve, we will be able to recuperate. And I'll show you some of the exciting work that we are doing in this particular area. When I started this work 3 years ago, my first postdoc, research associate, Brett Williams, who's seated out there, I sent him up to Charleville to collect plant material. And I get this nervous call from Brett to say that, you know what, plants are all dead. I think we have just wasted the money to come out there, spend the night and collect the plant material. So I told Brett, no need to panic. Put those into bags, I'll pick you up at the airport or see you back at the lab. By the time Brett returned to Brisbane, those plants were green, luscious, and they had resurrected. And I didn't have to convince him otherwise to work on this system, and he's been a pillar that has been working of great support in us trying to dissect the mechanisms that this particular plant adopts. So as I said, this particular plant, Tripogon loliformis, which I'm going to talk about, is a resurrection plant, and I'll demonstrate by a video just now what resurrection actually means. It can remain in this desiccated state for days, weeks, months and years. In fact, when I was at the University of Cape Town, I took a 10-year specimen from a herbarium and put it into a glass of water, and it resurrected within 72 hours. So that's the versatility of this particular system. Because it's a grass, most of our crop plants are actually grasses, the true cereals. There's an advantage associated with that. It's also diploid. Most of the resurrection plants, in fact, all the known resurrection plants are polyploid, which means that it's got many sets of chromosomes, making it very difficult to work with. This gives us an opportunity to do some very exciting reverse genetics, etc. So here you have a plant in its natural habitat, is exposed to a range of extreme conditions and seem to tolerate it quite easily. So it has to have some strategies or unique approaches that we could actually get some insight from, that we could utilize in improving stress tolerance in crops. Okay. Now, I'm going to show you a bit of structure associated with this particular resurrection plant, because my student Paraj Kabashi, who's seated in the second row there, has done some outstanding microscopy, and it's also to showcase the capability that we have at QUT through our Center for Analytical Research, where we've got some of the best microscopy facilities in Australia. So this is a transverse section through the leaf of tripogon. You'll notice that in its completely dehydrated state, it actually has folded. And these bulliform cells here lose their water, allowing the leaf to actually fold. But you'll notice that it's still green in color, and it has a pink or red outer layer. That outer layer is actually the abaxial part of the leaf, which is the lower surface that now has this film of anthocyanins that's actually protecting it from severe light radiation and preventing any further damage to occur to the actual tissues. Now, we also have a type of microscopy called an environmental scanning electron microscope, one of the few in Australia. The challenge with the resurrection plants is that if you've got a plant that's got just 5% relative water content, if you had to fix it, any fixative will always have some water in it. And the moment you put it into such a fixative, the plant resurrects. So how do you actually study it in its true desiccated state? And this is a powerful tool that we have used. So this is the adaxial, the top surface of the leaf, that we are seeing at around 500 times magnification. And this is the stoma, stomata, the trichomes, etc. But what I wanted to show you is this outstanding electron micrograph showing the leaf actually folding. And this is really the work from Paraj that demonstrates when this particular plant in its most dehydrated state actually takes on this type of conformation. This is from the apical part of the leaf, and this is from the lower part of the leaf. Okay, so this is, what I'm going to show you now is a video clip. Okay. Let's stop it there. So this is the work of Brett and Isaac, the students seated out here, where they took 1,500 photographs every 5 minutes from the time the plant was in its totally desiccated state. They re-watered it, and they then tracked how the plant responds. And when you take all these 1,500 photographs and put it together in this time-lapse video, this is what it looks like. So in effect, within 24 to 48 hours, this plant has made a full recovery and is able to now photosynthesize and literally get itself moving. And so that was really the beginning of trying to go ahead and dissect and identify some of the strategies this plant is adopting. And we have done extensive structural molecular work on this to get a good insight into what's going on. But I want to go back to the point that I made earlier on. Most plants, mesophytic plants and our crop plants, all fall into this category. When they are exposed to a stress, whether it is high temperature, low temperature, but particularly dehydration, they reach a certain point where they make a conscious decision that it is too difficult to hang in there, and they self-destruct. Now, if we could transfer a technology that allows us to reverse engineer the way the plants perceive the stress, and rather than convey a message of programmed cell death, convey a message of anti-programmed cell death or anti-apoptotic, we could actually, this could be a game changer. And there have been groups across the world that have been looking at it, but no one has looked at it in resurrection plants. So this is Isaac's work. Isaac is a PhD student from Kenya. What you're seeing here really is the use of Evans blue. If the cells have been damaged and the membranes are more porous and are no longer maintaining cell integrity, Evans blue will penetrate it, and actually the leaf will stain blue in color. And this is what he did by boiling the leaf, damaging the membranes, and that's what it looks like. But take a look at this. This is a hydrated 60% relative water content, 40 and 10% and less than 10%, and this plant is still maintaining the integrity of the cells. So their cells are actually not dead. And I believe that this is exceptionally phenomenal. Now, we have done, using what we refer to as high throughput functional genomics, and using some of the latest technology that allows us to sequence and find out all those instruments that I talked about in the orchestra that are switched on or playing at any one time. Brett has led this work, and we have got some absolutely remarkable data. QUT has just purchased one of the state-of-the-art Illumina sequencers. This actually involved the use of that platform, not the QUT one, but that type of platform to get this data. What you're seeing here really is the plant being dehydrated down to 10%, rehydrated, and this is in the shoots and in the roots. Okay? What we have noted is that if you take a look at all the genes that are associated with programmed cell death or pro-apoptotic, everything that's in red here means that it's downregulated. So there's clear evidence that when the plant is under stress conditions, it's not expressing these programmed cell death genes, which has validated our original hypothesis. But what about anti-apoptotic or anti-programmed cell death? These are genes or instruments that are indicators of that. And if you take a look at it, the green labels that you see there give us an indication that those anti-programmed cell death or so-called anti-apoptotic genes are actually expressed, which means that it's conveying the signal to be able to keep it alive rather than self-destruct. And you're probably familiar with the concept of senescence, which is where the plant conveys a message that it's time to actually break down all the systems and destroy the cells and allow the leaf or whatever to fall away. And we see the inducers of senescence downregulated, but those that delay senescence upregulated. So we have a manuscript that's currently being written up around this. But the bottom line here is that I wanted to give you a snapshot using some of the most sophisticated and high throughput and cutting edge technologies that are currently available. We were able to demonstrate and validate the observations that are made earlier on. Okay? There was also this brainwave that Brett and I had when we first started this work, and we said, well, if this plant has got all this interesting repository of genes and instruments, let's create a library of all of these genes that are switched on under drought stress or severe dehydration stress. Let's transform a model plant like Arabidopsis with them, and then let's select for those that are actually stress tolerant. And we call this complementation by functional sufficiency, and we used Arabidopsis as a model system. So what this entailed is that we grow Arabidopsis plants, wait till they flower. In this tub here, what we've got there is the agrobacterium containing these genes of interest. And then we go ahead and dip them in there. It's as simple as a floral dip. Collect the seeds from that, and we wait for them to grow, and that will be your T0 seeds, which means that they're transgenic, but they have just come out from that event. We self them, they become T1 generation, transgenic first generation, and we then go ahead and actually assess them for stress tolerance. And this is one of the selection plates showing that under antibiotic selection, all the other plants didn't grow, seeds didn't germinate, but this one here had some gene of interest. We've got some very interesting data on that, but I'm going to show you some data that I had obtained. One of my PhD students, when I was at the University of Cape Town, Delia Garwe, had obtained using a similar approach in my lab. So what Delia had done is that these are three different transgenic lines containing the gene XVSAP1, which came from another resurrection plant. Delia took these control wild-type plants and these three transgenic lines, and against conventional thinking, she took these and put them at 42 degrees Celsius for 12 hours. And then she took them out, put them back into normal growth condition, and this is what she got. It demonstrates to you the power of this complementation by functional sufficiency in identifying those genes that are functionally important in stress tolerance, and this is a classical example of that. I'm going to now switch gears to this area which I refer to as bridging the innovation chasm. A lot of this fundamental research that I hope I've got you excited about can very easily stay in publications, reside in journals, probably get excited about them, but really may not reach the marketplace. The DNA of QUT, and the thing that I enjoy working at QUT about, is that our focus is really on getting this to an industry outcome or to the marketplace where it could make a difference, whether it is for philanthropic purposes or whether it is addressing a specific industry need. And I'll give you an example of how we are doing that. Now, you are probably aware that I have a project that's actually funded by the Queensland government around addressing some of the major challenges the tropical pulses industries are experiencing, particularly chickpea and mung bean. Now, if I can set the context around this, there is this, particularly globally, that there's an increasing demand for a vegetarian source of protein. Now, particularly in the Indian subcontinent, we find that there's an absolutely growing need for this. And we are finding that the more affluent people are preferring to actually get a plant-based protein as compared to an animal-based protein, whether it is through vegetarianism or otherwise, but that's a growing trend. Now, if you take a look at the opportunity that this presents us here in Queensland specifically, because a lot of chickpea and mung bean is actually grown in Queensland or in even northern New South Wales, we notice that while India produces a significant amount of these tropical legumes, particularly chickpea and mung bean and other legumes, they still import around 5 million tons per annum, which means that we in Australia could actually satisfy this market niche. But the demand has been so insatiable that almost 98% of what we grow in terms of chickpea, for example, gets imported by India or gets exported to India from here. So how do we actually grow this economically so that the economy of Queensland and this particular industry could benefit from it? So we turned to the industry and we said, well, what are the major challenges that you're experiencing? And it's not rocket science that drought, disease, etc., were high on their priority list. In fact, over the past 2 years, the chickpea industry has actually lost almost 40% of their crop to a pathogen called Botrytis cinerea. And I'll show you how we are helping to deal with some of these. Okay? Now, chickpea and mung bean, in most cases, should not be something that you would not have encountered. This is two very valuable sources of dietary fiber, minerals, and in addition to that, a very important source of protein. So even if these photographs don't ring a bell within you, I'm sure the following would. These are just some of the products that you get from chickpea, and I'm sure you consume hummus, a nice chickpea curry, chapati, falafel, and obviously chickpea powder. And these are just some examples of products that I'm sure many of you encounter in your day-to-day activities. How about mung bean? I'm sure some of these are familiar to you. The sprouts, papadum, and your noodles, and the mooncake actually is renowned for containing mung bean. Now, let's take a look at this from a helicopter view. If we look at this industry within Queensland and take a look at where most of our product go, and this is data that I've obtained from Pulse Australia, you'll notice that the majority of our product, in this case the chickpea, is actually exported to the Indian subcontinent, which includes India, Bangladesh, Pakistan. Okay? But we do have 7.8%, at least during the period when the assessment was done, went to the Middle East. Now, what you would notice here is that the demand, and this is just to reiterate the point I made earlier on, the demand to India has grown significantly and it's continuing to grow. So we clearly can actually align a lot of the economic growth that we see to this opportunity that exists in that part of the world. Now, what does this translate to in dollars? Okay? This is an industry, as you could see, from 2011-12 that was around $350 million, has actually grown to close just over 500 million. And we believe that this opportunity is actually even greater if we can help deal with some of the issues and challenges that this industry is experiencing here in Queensland and Australia at large. And it's a very similar picture for mung bean. The majority of our product is actually going to the Indian subcontinent. Now, we believe that in order to be able to help address the growing demand for food, but in this particular case I'm saying protein specifically, we need to be smarter. We need to be addressing the food security issues by what I refer to as using smart agricultural approaches. Okay? And I'll give you an example of how we not just look at using the biotech approach to deal with these issues, but also looking at a range of other strategies to be much more smarter in terms of what we want to expect from this. Now, I'm going to talk about a novel chemical pre-treatment that we are working with an entrepreneur and the impact that it's having on root architecture and particularly stress tolerance. I'm also going to talk about the fact that using this anti-apoptotic or anti-programmed cell death approach that we see in the resurrection plant, we believe that we can help address the drought tolerance as well as disease resistance. Because this bacteria, this fungus here, Botrytis cinerea, is what we call a necrotrophic pathogen. If a plant is under stress and if there's some destruction of the cells because of that and a spore lands on it, it proliferates. If you're able to keep the cells active and alive, you can be assured that this will actually result in botrytis resistance. And some groups have already shown that in model systems. But I do believe that we need a few game changers. If we want to get our products out into the Indian subcontinent, what are some of the major issues the Indian subcontinent is addressing or dealing with? One of it is anemia. There's insufficient iron in the diet of an average Indian or average person living in the Indian subcontinent. So we maintain that if we could look at enhancing the iron content and making it more bioavailable in these products, our products would be accepted into those markets much more easily than products that are coming from other parts of the world. So this is what we refer to as a smarter way to deal with not just food security, but health issues to what we call nutritional security. And there's also a breeding component that we're involved in, and I will talk a bit about using computer modeling to be able to predict how we can best grow certain crops with certain genotypes in certain regions. Okay, so let me show you firstly some of the partners that are involved in this, including Pulse Breeding Australia, the Australian Mung Bean Association. This is funded by the Queensland government, but these are other key stakeholders that we interact with. This is the team that's behind this particular work. 3 postdocs. In fact, all of these members are here. 3 research assistants, 2 PhD students. Professor TJ Higgins, who is at the CSIRO, has been very instrumental in helping us to set up our transformation system. And Rex Williams, who is the science leader in DAF, Yash Chauhan, as well as Cole Douglas, who are all part of the team. All of them are listening in currently, live streaming from Toowoomba, Kingaroy, as well as Warwick. So they are participating by observing and watching, and I'm sure that if they have any questions, they will be getting that through as well. All right, let's talk about this novel chemical pre-treatment, which I believe is quite a game changer. Okay? Now, there's an entrepreneur called Mark Burns who approached me 18 months ago with this novel chemical pre-treatment that has been shown to enhance root architecture or fine root growth. And in doing so, he has been using this for the last 25, 30 years. And Mark is also streaming in live to listen to this presentation. He was able to revegetate disused mine sites across Australia. So we said, well, if this is resulting in such a major impact in generating stress tolerance, let's see whether we could actually get to the bottom of what this is doing with some of the crops that we are working with. So we have gone ahead and looked at the impact of this particular chemical in vitro, in tissue culture, in glass house, which is basically based out in this particular block on level 9, and in field trials. And we currently have field trials out in Gundawindi as well as in Roma, and we have just completed field trials at Hermitage, which is based in Warwick. Okay? So what exactly is this particular product doing? Now, if we take this product, which we call ATW1124, it's a natural chemical compound that's produced naturally by the plant, and it's also fairly cheap. If you take the seeds and just treat it without any treatment, and this is where you treat it with water, and here you use 5 millimolar ATW1124, you don't have to be a rocket scientist to figure out that it's having a significant impact on root growth. If we take a look at mung bean, we see exactly the same effect. In the various treatments that we had undertaken, the root architecture has changed significantly, much deeper, more robust root growth. Now, we went ahead and set up a glasshouse trial, and here you see what is the control of a chickpea plant, and this is one that has been treated, and we have noted consistently that there's a significant enhancement of root growth, and there's been no penalty on the productivity as well as the aerial parts of the plant. However, it does, with deeper rooting system, you would expect that it will give it an enhanced ability to absorb nutrients and to survive severe drought conditions. Now, I couldn't help but put this particular slide in. This is the work of Michael Dott, and Michael is seated at the back there. We have been using what is called X-ray computerized tomography, where we grew these mung bean plants that have been treated with ATW1124 in vertical tubes. And without disrupting the plant, we went ahead and did a CT scan of these roots. Okay? And if you take a look at this, and this data is just a couple of weeks old actually, with just the water treatment, that's what the root architecture looked like. And be mindful that this is now sitting in a tube that's probably about 5, maybe about 10, I would say about 8 or 9 centimeters in diameter. Take a look at the 5 millimolar treatment, the significant enhancement of root growth, and the 10 millimolar. So we believe that this particular chemical treatment, if we can demonstrate its effect in the field trials, we have a product that the farming community could utilize almost tomorrow. But the challenge now is to obviously see whether the effects that we are seeing in tissue culture in the glasshouse is actually translatable to what's happening in the field. And in doing so, we are looking at a unique strategy to enhance root architecture and root growth and hopefully result in stress tolerance. The other approach that we have used is really a biotech approach, using genes from the resurrection plant to enhance stress tolerance while at the same time improve disease resistance. And this project has just been around just about a year and a half in existence. So what I'll show you is an update on where we are at present. This work is spearheaded by Sudipta Das-Bomik, who is seated, who's present here as well, and Alan Cheng, who is the research assistant. Essentially what we do is that we take chickpea seeds, imbibe them in water, split them in two, expose them to the genes of interest which is present in the agrobacterium, go ahead and select for your transgenic material, and you could see the robust growth during regeneration. Because it's difficult to root chickpea, we go ahead and micrograft them onto rootstocks that are non-transgenic. And it's a fine art. And I should tell you that Sudipta and Alan does on average 1,000 transformation seeds that are transformed per day, and they do around 3,000 per week, and so far they've done over 68,000 seeds. And you could imagine the high throughput. This is what you do in industry to be able to get a positive outcome. And the graft rate, okay, successful graft rate is around 90%. That's how fine-tuned it is. If we are able to get the seed from this material, and all of these here that you see have been validated to be transgenic material, because you can do that by carrying out a polymerase chain reaction and look for your gene of interest. If we can get a seed from this plant here, using some, again, really recent technology, and this is in collaboration with the University of Western Australia, we could get up to 8 generations in a year. We don't need 8 generations, we just need to get up to generation 3 so that we could go ahead and test this in the glasshouse and hopefully in the field. So you could see what I'm trying to demonstrate is that there's an opportunity to get a product to industry sooner than expected. Now, I've already mentioned this value adding around biofortification, but I think we underestimate the importance of iron and the impact that anemia has across the globe. In fact, if I take a look at a figure that the World Health Organization had put up, what you're seeing here is that if it's dark orange in color, it's actually a really severe issue. And if it's moderate, like what you see in most parts of Europe, this is what it looks like, and this is where there's a mild issue around anemia, which is really iron deficiency that's becoming a growing health issue. Now, the point I wanted to make here is that we need to be smarter in our agricultural activities in helping to address this in the kind of exercise that we are undertaking. While we might think that this is largely a developing country issue, this particular map here and this graphically represents the fact that it is not just a developing country issue, but it's also an issue in the developed world as well. Okay? And the last pillar that I will talk about, which demonstrates the smart approach to agriculture, is, all right, we improve the genetics of the plant, we know what sort of traits we have, we know the kind of environments we want to expand our cropping in, but how do you now put in a management practice that makes sure that you have a successful crop? Yeah, this work is led by one of our team members, Yash Chauhan, and he uses computer modeling to pull all of these things together and to be able to tell with a fair level of accuracy where we could expand certain crops to. So even before the farmer has put seeds out, we could essentially, using crop modeling and looking at the genotypes that we have, we could determine what sort of success rate you could have. And this is not rocket science, because Queensland has actually led this work together in partnership with CSIRO, particularly in the sorghum crop, and that's how we've been able to grow the sorghum industry in Australia. But we are hoping to pick up the learnings from them, and this will be the first time it will be applied to a leguminous crop. And we believe that this will provide a tangible outcome for our farmers to be able to address. And not just that, we could reach out to other cropping systems that are present in Asia and Africa, where we could model what sort of genotypes are needed to survive in such environments. And we're already developing collaborative projects with East and Southern Africa, as well as in parts of India. Now, I'm putting this all back again as I'm winding up this talk to emphasize to you that this is a really complex issue to deal with. And this afternoon, you have given me an opportunity to what I call give you a perspective on an area that I'm very passionate about. And I believe that there is no single approach or track that's going to result in the kind of security that we are looking for. But I think it's going to require a plethora of approaches and skills to be able to help address it. And that's where I think the multidisciplinary approach of including breeders, including crop modelers, including molecular biologists, biotechnologists, helps us to find what is the best way we could help deal with an issue. It's not the person who speaks the loudest or the person who has the most influence, but it's the best idea which is really what should survive. And hence, helping to deal with issues like population growth, land degradation, dealing with the food security issues, and looking at future crops in terms of how we can best help deal with these issues is going to be pivotal in us being able to help address the challenges that we have. I could not help but also provide you with a personal experience that I've had. While I've presented to you all these high-tech approaches on how we can deal with the issues associated with trying to feed the next generation and also creating crops for the future, I worked with a community in South Africa, in the eastern part of South Africa, where there were actually around 500 farmers, mainly women, with young families in a formerly war-ravaged area that were growing indigenous crops. And I looked at them and my heart actually went out for them, because they were literally working and using all these local vegetables, trying to get it out into the local markets, and some of them Western society had not even been exposed to. So what I did is that, in my role as CEO of South Africa's National Innovation Center for Plant Biotech, I got the local academic, who actually is a plant breeder, to identify the karyotypes that would probably produce a consistent product. Okay? This particular product here, if you've been to Southern Africa, is called a madumbi. It's a local term, and it's probably in most parts of the Pacific region is called a taro, but this is specifically found in Southern Africa. What we had done is that we took the youth from this community and allowed them to create a co-opt so that they were able to manage the business associated with trying to get this product out to the broader marketplace. I invited the CEO of Woolworths South Africa to visit this community. To cut a long story short, their products are now available in every single Woolworths store in South Africa. It was not high-tech, it's a low GI product, and an average South African home, if you've been to South Africa, will always have a madumbi if you want an afternoon snack. So I leave you with this African pot, a three-legged pot, with the objective of emphasizing the necessity for these partnerships. We cannot work as an institution in universities or otherwise in isolation from what the needs are out there. I believe we need to keep a foot and a finger in the pulse of what's happening in the community. And these issues around health issues around anemia and other similar issues need to be addressed. We also need to work closely with industry to find a vehicle to bridge that innovation chasm so we could actually deliver tangible outcomes to these communities. This would not have been possible without very strong partnership from a range of funding agencies, in partnership with CSIRO through TJ Higgins, Mark Burns, through Plant Solutions Australia. But most importantly, this is my team here at QUT, the Abiotic Stress Group. There's 12 of us there, and we come from 9 different nationalities. That itself represents, the diversity of what the world, the marketplace has become, and what university environments have become. So with that, I really thank you for your attention and welcome questions.